We are members of Professor Kabin's research group at Huazhong Agricultural University. Herein, we will introduce our recent work submitted to iMeta, entitled Cultivar-specific preference of bacterial communities and host immune receptor kinase modulate the outcomes of rice-microbiota interactions. This study designed two versions of gnotobiotic growth bottles suitable for the growth environments and conditions of rice, which are used for investigating rice microbe interactions. Version 2 facilitates the exchange of nutrients between the interior and exterior. Microbial inoculation significantly promoted crown root growth of rice cultivar MH63, and no significant difference in this promotive effect was observed between version and version 2. However, under version 2 conditions, soil microbes remarkably enhanced the plant height of above-ground parts. Additionally, the gnotobiotic culture devices are easy to upgrade and modify. Our findings demonstrate that the same bacterial inoculum exerts beneficial, neutral, or detrimental effects on root growth across different rice cultivars. An imbalance in the absolute abundances of detrimental and beneficial bacteria in the roots is associated with the promotive or inhibitory effects on root growth in the two rice cultivars. Additionally, the conserved receptor-like RLK, OSFLS2 is essential for preventing microbiome dysbiosis. This study found that the gnotobiotic growth system can simulate the root growth environment of rice under natural conditions. The promotive effect of inoculated soil microbiota on root growth of rice cultivar MH63 is manifested in crown root elongation and increased lateral root density, and the same microbial community exhibits significant differences in its effects on root growth among different rice cultivars. This study investigated the assembly of bacterial communities colonizing the roots and rhizospheres of rice cultivars MH63 and KIT. Our results showed that bacterial community assembly conforms to the assembly rules under natural conditions. A small number of core bacteria occupy root and rhizosphere niches, and these colonizing bacteria exhibit low species overlap across different rice cultivars. From a logical perspective, their assembly is a stochastic process, which is not entirely consistent between MH63 and KIT. The dispersal process predominates in MH63, while the drift process is predominant in KIT. Based on the assembly rules of root colonizing bacteria, this study isolated 22 cultivable bacterial strains covering 4 major phyla in the roots to construct a synthetic microbial community, SynCom22. Subsequent screening of these 22 strains, with half removed, yielded a simplified SynCom consisting of 11 strains, SynCom11. Rice cultivar MH63 was inoculated with SynCom2 and SynCom11 separately. The results showed that a small number of core bacteria could recapitulate the root growth promoting effect of complex soil microbiota, thereby reducing the complexity of the research. Inoculation experiments with individual strains and SynCom11 both demonstrated that the same microbe exhibits preference in its root growth promoting effect across different rice cultivars. Further analysis of the absolute abundant colonizing bacteria revealed that the colonization level of beneficial bacteria specific to each cultivar may determine the effect of the microbial community. When beneficial bacteria dominate or exhibit higher colonization levels in the roots, the microbial community exerts a beneficial effect on the plant. Beyond characterizing microbial composition, we sought to understand how a beneficial microbiota promotes root growth in the presence of host immunity. After screening a KIT background RLK mutant library, we identified 4 mutants that subverted the RGP effects of NSM, namely, including OSFLS2 and OSERK1 mutants. We prioritized OSFLS2 and OSERK1 for further analysis because FLS2 is a classical PRR that recognizes flagellin and OSERK1 is one of the central regulators of plant immunity. In germ-free soil, the root length of OSFLS2 and OSERK1 mutants were comparable to those of the WT plants. While NSM enhanced WT root length by 18.5%, it inhibited root growth in OSFLS2 and OSERK1 mutants by 11.8% and 18.9%, respectively. Analysis of the root bacterial communities in mutants revealed that the diversity of endophytic bacteria in FLS2 mutants was significantly lower than that in KIT and SERL1 materials. Further analysis of the differential bacteria in the rhizosphere and roots of FLS2 showed that Rhodocyclaceae and Burkholderiaceae were enriched in OSFLS2 endophytes, while Pseudomonadeaceae and Enterobacteriaceae were reduced. Root counting also indicated that the number of CFUs per gram of roots in the two mutants was significantly higher than that in the wild type. These results demonstrate that the deletion of key immune receptors leads to microbiota dysbiosis, characterized by 1. Impaired plant growth, 2. Altered microbial community structure, 3. Significant changes in endophytic bacterial load. Transcriptome analysis of KIT and FLS2 plants grown with and without microbiota revealed that a large number of differentially expressed genes, DGs, exhibit opposite expression trends between KIT and FLS2. These DGs are mainly enriched in defense-related functions, indicating that the microbiota can modulate the plant's defense responses through receptors such as FLS2. OSFL plays a crucial role in microbiota-mediated regulation of host gene expression. On the basis of these results, we propose that the rice microbiota relationships are gated by cultivar-specific preferences of the bacterial microbiota and host immune receptor kinase, which provides a useful framework for crop microbiome engineering in the future.